All right, everyone. So I am going to be going over the cell structure, essentially, and all the organelles within the cell. So, if you will, welcome to the cell. This is inside of a Trader Joe's grocery bag is, in fact, a cell, believe it or not. So, we have the internal cytoplasm here, and then you can see on the edge, this is essentially the cell membrane. So, peripheral proteins, glycoproteins, cholesterol, channel proteins, and then again, we have our, phospholipid bilayer. So, it stretches along the whole thing, and then this would be the bottom. So, again, phospholipid bilayer, I didn't want to draw out a billion little dots. So, yeah, so that's kind of the, overall structure. And then we would have the cytoskeleton kind of supporting everything, which imagine there's kind of just intermeshing, area enclosing here and kind of supporting everything as well, you know, additional to the organelles. So let's get into the organelles. So, good starting point, I feel like, is with the powerhouse of the cell, mitochondria, which I've, carved cristae, which are supposed to be internal folds, but, I did my best here on this carrot. That's not a carrot, it's a mitochondria. Synthesize ATP, hugely important. Again, cell powerhouse, I feel like, is the kind of known meme, but that's exactly what it is. That's a good starting point. So, yep, that's one organelle. Another organelle that we can look at is the Golgi complex, which is not this set of bananas, definitely, but in fact, a real Golgi complex. And with this Golgi complex, it's made out of cisternae, which I did not know how to visualize really on a banana. That's not a banana, it's a Golgi complex. And this Golgi complex essentially helps finalize protein peptide synthesis and synthesizes carbs, and also produces Golgi vesicles, which these grapes may or may not be grapes, but may also be Golgi vesicles. And these essentially, are the finalized proteins. These will float around the cell, move about, do whatever they need to do. And one type of that, which I happen to be holding, is a lysosome, which is essentially a white blood cell. It moves around the cell, consumes bacteria, other foreign, substances, essentially. And again, is made by the Golgi complex itself. So just one of the vesicles that can pop off. I mean, again, I probably should have started with the nucleus, but we have our beautiful nucleus here, which is not a stress ball I use to strengthen my fingers for rock climbing. It's definitely a nucleus. And we have the nucleolus at the center here, which we know is in charge of making ribosomes. You can use essentially the whole, if I'm pretending this is the nucleus, the whole outer layer here that goes around can be viewed as the nuclear envelope. So that's, again, the nucleus, and attached to the nucleus, continuous with it, we have the rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, we know the rough houses ribosomes, which, here's some ribosomes floating around freely in the cell. The ribosomes on the cell, so, this is, again, not a raspberry. This is a rough endoplasmic reticulum. So these ribosomes, again, made by the nucleolus, can pop off, float around the cell accordingly. Then we have the smooth endoplasmic reticulum, which is continuous with the rough. So not the nucleus, but with the rough. Again, not a chocolate with a puppy's face on it, but that's smooth, the endoplasmic reticulum. And one of the interesting things about smooth endoplasmic reticulum is it doesn't have ribosomes, but it does detoxify the cytoplasm. So also an important role there. And then we also have peroxisomes, which are actually produced by the endoplasmic reticulum, so kind of a good transition there. White blood cells, not a candle that I burned the living daylights out of on the back, but definitely a white blood cell. And again, similar to the lysosomes, will move around injecting foreign material, but different kind of origin area, different area where they're produced. And then that leads us nicely into our last organelle, which are centrioles. Again, microtubules, kind of banded together, perpendicular cells, bundles of them. And these are essentially used to help with cell division. So, when the cell is breaking apart, these will kind of come into play. They're proteins, so they can kind of help in that cell division. And if you were to look at it, ideally, I believe the picture in the text kind of has it viewed like this. So, definitely not three pencils together. So yeah, that's kind of the cell construction. Hopefully that helps you guys. It's kind of helped me talking through it. all this food will be put to good use. I will not be eating the candle. Thank you.